Greetings again, my students. We're going to continue with a bit of statistics to help us go on to the other statistical aspects of plant breeding. To summarize what we talked about before, we had uh, indicated the statistics that can be used in informing about the accession lines that have been identified for further study. And so the question is, when should one use covariance? And the answer, to help you, is we use it when we want to see how much two or more trait values change in relation to each other. In statistical terms, the equation is given as follows, and E is stands for the expectation, and this can be calculated as the summation divided by the number of observations as seen in this equation. Notice that there is a difference for the population covariance and the sample covariance. The sample covariance being n minus 1. Generally speaking, covariance is affected by the change in scale. In other words, if all the value of one variable is multiplied by a constant, and all the value of another variable are equally multiplied by a similar or different constant, then the covariance is changed. Now, generally, a covariance, as one can see on the figure that is on the left, we have similarity with the other graphings that we will look at for correlation and even regression, but they have different meaning. On the left here, I have put the basis of comparison between covariance and correlation, and they are self-explainable. But to emphasize, covariance indicates the extent to which two random variables or two traits change in tandem. Correlation is simply how strongly two variables are related. So with covariance, you note that it is the extent to which two traits change in tandem, in connection with each other. Whereas in correlation, it is the strength between two traits. So covariance is simply a measure of correlation. And a change in scale will affect covariance, but it does not affect correlation. Now, what about correlation? Now, correlation can be said to be the process of establishing a relationship or a connection between two or more measures. Let's take just the issue of height and weight. You can see that we have the row that says height, and we see in that same row, it is compared against the columns that say height and weight. Height and height compared to itself, then height compared to weight. And then in the second row, we see weight compared to height and weight compared to itself. Now, if we measured the Pearson correlation, we see that 0.000 means that the R is significant at 1%. All right? Now, these data here are an example of some of the work that is very similar to a tool I used. These are not my data, but a tool that I used called the X-ray fluorescence analysis, where you get an accession which can generate a lot of mineral micronutrients in a single run. And indeed, there can be as many as the ones you see in the column, in the top row here. And then what I did when we get this data, this is just for a single accession, for example, where I wanted to capture ionic variation, mineral micronutrient density within the accession. Then you see, within that given accession, for example, is where you can have the row compared by the column. And the figures, as they are shown here, especially the ones which are underlined or underscored, they show that these are significant correlations where, for example, you have the fluorine associated with bromine and so on and so forth. And you have all of these that are significant, meaning that there is a strong relationship with these minerals as they exist in the plant tissue. So correlations, as one can see this very quickly, are those that can show a strong relationship, positive or negative, especially if one can look at the correlation coefficients as shown in this box on the bottom right. So in statistical terms, the estimated correlation can be estimated by this equation here. So covariance is nothing but a measure of correlation. On the contrary, correlation refers to the scale form of covariance. Let's not get confused about it, but just get the concept of correlation as is distinguished from covariance. So correlation, said another way, does determine the degree to which two or more random variables move in tandem. During the study of two variables, if there has been the movement in one variable, it is similarly reciprocated by an equivalent movement in another variable. And we'll see this as we go to the forward slides. So in some way or the other, then the variables are said to be correlated. Got to emphasize here that correlation is dimensionless. It is a unitary measure of the relationship between two variables. It is unlike covariance. In covariance, the value is obtained by the product of the units of the two variables. Both measures only linear relationship between two variables. When the correlation coefficient is zero, covariance is zero. Further, the two measures are unaffected by the change in location. So the conclusion here is that correlation is a special case of covariance which can be obtained when the data is standardized. Now, when it comes to making a choice, which is a better measure of the relationship between two variables, correlation is preferred over covariance because it remains unaffected by the change in location and scale, and also can be used to make comparison between two pairs of variables. And so plant breeders will generally use correlation in reporting the strength of a relationship between the accession variables. There are four measures of correlation. We have scatter diagram, we have product moment correlation coefficient, we have rank correlation coefficient, and we have coefficient of concurrent deviations. But suffice it to say, we really do not have to worry a lot about the issue of the various types. But let's just summarize the differences between
Before we
correlation
go any
and
further,
regression.
let's focus on the relationship between correlation analysis and regression analysis. There is a single expression that sums it up nicely. Correlation does not imply causation. Let's spend one more minute on that. First, correlation measures the degree of relationship between two variables. Regression analysis is about how one variable affects another or what changes it causes to the other. Second, correlation doesn't capture causality, but the degree of interrelation between the two variables. Regression is based on causality. It shows no degree of connection, but cause and effect. Third, a property of correlation is that the correlation between x and y is the same as between y and x. This you can easily see from the formula, which is symmetrical. Regressions of y on x and x on y yield different results. Think about our example with income and education. Predicting income based on education makes sense, but the opposite does not. Finally, the two methods have a very different graphical representation. Linear regression analysis is known for the best fitting line that goes through the data points and minimizes the distance between them, while correlation is a single point. All right. Hopefully, this short video clears some doubts you had. Stay tuned.
So we've seen that in case of a rather high correlation, then we can get into the question, how much change in Y do you see with 1 unit change in X? So when we have a high correlation, highly significant, then that question may begin to hint on the possible cause effect, which takes us into regression. That question can be answered by the regression coefficient. As we've already seen, a measure of relation between X and Y, but expressed as a proportion, the variance of X, expressed as a proportion of the variance of X. And it is usually denoted by B, which is a slope in our graphical representation. So that B, the slope, is the change in the value of Y when X as an independent variable is 1 unit higher. In other words, as X changes 1 unit higher horizontally on a graph, then there is a change in the Y. It causes a change in the Y on a vertical scale. And the regression coefficient can be calculated as seen in that formula. And it can also be either negative or positive, depending on the sign of correlation or covariance between the trees. The regression coefficient is very important because we can regress the progeny yield on parent yield and then be able to estimate the heritability for yield. And we'll get into that a little bit later. So we have the single regression. It's a mathematical equation. It has a model, and it includes only 2 variables. Compare with correlation. Correlation, you can compare several variables. In a single regression, simple model, you have only 2 variables, 1 independent and 1 dependent. So the dependent variable in most cases is the one that is being explained by the change in the independent variable. It's the one which is used to explain the variation in the dependent variable. All right? Now, let's remember our augmented designs that are used in plant breeding. And you have those many where you have the 4 accessions there, as you can see. And so as we go on, let's look at how the issue of the correlation is associated with other things when you are looking at the genotypes as the independent affecting some of the growth responses. And I would like that you go back to your notes and read this particular slide, and you see other aspects that are brought in with respect to the important traits that contribute to variation. Traits that contribute to variation. And then we have the molecular characterization, a walk on the inside. I like to call it the genotypic walk. You see, where you are now looking inside the cell, inside the nucleus, inside the DNA, then coming outside to see the trait characteristic. And we'll not spend time on this because we will be getting into the plant biotechnology in the next year of your final year to look at what that means. If we go back to our diagram before, remember on the left for determining the genes that contribute to shortness on the left tail and the many genes that contribute to tallness on the right side, we see that we can graph that variation across several people. Using the genomic analysis, we can analyze using SNP markers the variation among these individuals' genomes to begin to see how they actually vary in a molecular context. So such information obtained can simply be defined as a DNA position of information that needs to be transcribed into RNA, subsequently translated into a polypeptide chain with physiological function. This takes us to the field of what is referred to as genetic sequencing. The SNP sequences can show a variation among accessions, similarly as genome analysis can show the variation among the few genes of the short people and across to the genes of most people, across the genes of the tallest people. So it is in this field of sequencing called genotyping by sequencing, or GBS, that is a method that discovers SNPs, single nucleotide polymorphisms, in order to perform genotyping studies. And such studies are, for example, genome-wide association studies, GWAS. And this is just a quick indication of this, but we really don't want to go into that detail. But where we get a PCR done, and then it is looked at in terms of the sequence, which can begin to distinguish a variation based on the SNP panels that will indicate which uh, uh, particular genotype is different from the other. And so we will stop at that and we'll continue with part 4, where then we can revisit the issue of the reproduction as a window for crossing. Thank you very much for listening.